Today we are going to study about the histology of ganglions. The ganglions, as we have already studied, the ganglions are the aggregations of cell bodies of the neurons. They are the aggregations of cell bodies of the neurons which can be seen outside the central nervous system or in relation with the peripheral nervous system. It can be seen in relation with the peripheral nervous system or outside, we can say outside the central nervous system. So these ganglions are of different types. We are going to study about the histology of the ganglions. Histology of the ganglions. It is the aggregation of cell bodies of neurons outside the central nervous system. That means a neuron have got, majorly a neuron have got its parts like central portion which is named as the cell body or the cell mass, and there are small processes arising from the cell body or the cell masses, or they are reaching towards the cell body, they are the dendrites, and a single set of fibers which are arising from, and that is the axon. So this is the common structure of a neuron. So the aggregations of cell bodies of neurons which can be seen outside the central nervous system is known as the ganglion. The ganglia are of two types. They are sensory ganglion and the autonomic ganglion. Sensory ganglions are present just outside the central nervous system. Sensory ganglions are present outside the central nervous system, and neurons of the sensory ganglia, they carry impulses towards the central nervous system. That is the speciality. It is present outside the central nervous system, and the neurons of the sensory ganglia, they carry impulses towards the central nervous system. Examples are the dorsal nerve root ganglia of the spinal nerves and the sensory ganglia of the cranial nerves. The dorsal nerve root ganglia of the spinal nerves means the dorsal nerve root ganglions, they are present in relation with the uh, spinal nerves from which are emerging from the spinal cord. At the emergence or at the position of origin of spinal nerves, it possesses two rami, the ventral rami and the dorsal rami, or the ventral root or the dorsal root. They are joining together to form a single spinal nerve. So when it is arising from a spinal cord as two different various roots, the dorsal aspect, which is named as the dorsal nerve root, and the ventral aspect, the branch is known as the, or the division, the starting root is known as the ventral root. So the posterior or the dorsal root have got a thickening. That thickening is known as the dorsal nerve root ganglion. Like that, the sensory ganglia also present in cranial nerves like trigeminal nerve, vestibulocochlear nerve, glossopharyngeal nerve, vagus nerve. All these things have got sensory ganglion. Majorly, it supplies the or it acts as a sensory in function, or it supplies the regions which receives the informations towards the central nervous system. So that is a speciality of sensory ganglion. And in case of the autonomic ganglia, they majorly supplies the smooth muscles and glands. So autonomic ganglia, especially the speciality is that the parasympathetic and sympathetic ganglions can be seen under the category of autonomic ganglion. Parasympathetic and sympathetic ganglion, already the parasympathetic supply and sympathetic supply we have studied in case of the glands when we have discussed. So the secretomotor pathway or the secretory function, that is with the help of autonomic nervous system. So the parasympathetic and sympathetic supplies for the secretory function for the glands, and also the vasomotor or vasoconstrictor, vasodilator. Vasomotor means the blood vessels. The smooth muscles of the blood vessels, on the walls of the blood vessels there are the smooth muscles that we have studied in the histology. So the smooth muscles which are present in the blood vessels, they are supplied by means of the autonomic nervous system, so that this will help for the vasodilatation and vasoconstriction. That means the contraction of the blood vessel and the relaxation of the blood vessel. So these are the importance of the ganglions are present in relation with the central nervous system and peripheral nervous system. So first we will study about the autonomic ganglia. The autonomic ganglia, that is majorly supplying the smooth muscles and the glands. The autonomic nervous system shows two types of neurons. That these terms are familiar already: preganglionic and postganglionic fibers. Preganglionic means the fibers are arising from a nuclei which is present in the central nervous system, and this is passing towards a ganglion which is present in the peripheral nervous system. So the fibers which are receiving at the ganglion is known as, that is, which is present before the ganglion, which are receiving at the ganglion, is known as preganglionic fibers, and the secondary set of fibers which are emerging from the ganglion is known as postganglionic fibers. So in case of autonomic nervous system, it shows two types of neurons: preganglionic and postganglionic neurons. Example for sympathetic ganglia are superior sympathetic ganglion, inferior sympathetic ganglion, all are the examples for it. And also aortico-renal ganglia, inferior mesenteric ganglia, all these are controlling the blood vessels, the flow of the blood vessels. Autonomic or sympathetic ganglion, how it appears under the microscope is like, they are, this is the diagram showing the autonomic ganglia. The speciality of the autonomic ganglia are, they are multipolar neurons. That means in this diagram we can see a cell body possesses a number of projections. Cell body have got a number of projections. That projections are the polar, it is mentioned as the polar. So multipolar means two or more projections can be seen in a neuron. That's why it is multipolar cells. These projections represent the dendrites, and there in between there is the presence of fibers, discretely formed bundles. That means the sparsely arranged fibers can be seen in between the neurons, and the nucleus is present eccentrically. Eccentrically means not at the center. In this diagram we can see the single neuron have got nuclei with nucleolus in it, that is present eccentrically, either towards one side, not at the center. It is rounded nucleus, but it is eccentrically placed nucleus, and the nuclei can be seen with satellite cells. The satellite cells are the small cuboidal cells that surrounds the neurons in different peripheral ganglions of the peripheral nervous system. These cells, they are the cuboidal cells that surrounds, the neurons, and it provides structural support and the metabolic support. They provide structural support and the metabolic support to the neuron that they surround. They insulate the neurons and provide efficient metabolic exchange. So that is the function of the satellite cells. The satellite cells can be represented by small dots uh, at the cell walls. So that the speciality is that in this case, in the sympathetic or the autonomic or parasympathetic ganglion, there is a presence of only a little number of satellite, less number of satellite cells can be, supporting cells can be seen, and the neurons are spatially arranged. In between the neurons, there are discrete bundles of the the fibers, nerve fibers can be seen. So, and also there are the blood vessels can be seen in forms of sinusoids. So these are multipolar neurons. There are the myelinated or non-myelinated nerve fibers can be seen in between the ganglions or in between the cell bodies, and there is a presence of satellite cells. Less number of satellite cells can be seen in relation with each neuron, and there is an eccentric nucleus with the nucleoli. So these are the features which can be seen in relation with the, see, this is how it appears under the microscope. We can see here the neurons the cell bodies of the neurons can be seen in between. There is the nerve fibers, and this nuclei can be seen towards either side, not at the exactly at the center, so that eccentrically placed nuclei. And this blue dots represents the satellite cells. Blue dot can be seen in relation with this. It represents the satellite cells. So these are the features. These are the features which we have to study under the autonomic ganglion or the sympathetic or parasympathetic ganglion. They are multipolar, irregular shaped, smaller and scattered neurons. The nuclei of neurons are large and with prominent nucleolus and eccentrically placed in the cytoplasm. Only few satellite cells are present around the neurons. Neurons are separated from each other by discrete bundles of nerve fibers. These are smaller than the sensory ganglions. So these are the features which we have to learn under the microscope. Next is the spinal ganglion or the dorsal nerve root ganglion. The spinal ganglion, dorsal nerve root ganglion, otherwise it is the sensory ganglion. So not like the sympathetic ganglion, it is either pseudo-unipolar or unipolar. Pseudo-unipolar or unipolar cells can be seen. Pseudo-unipolar or unipolar cells can be seen. That means with a single projection. A single projection only can be seen in relation with the cell. So the speciality is that there is a collection of cell bodies can be seen in bundles, or in between this there are large bundles of nerve fibers. The collected bundles of nerve fibers can be seen in between the collection of cell bodies that will partially separate this. So this is the diagram showing. Here we can see a single unipolar cells with the nuclei. The satellite cells can be seen. They are very much visible and they are well defined, and a number of satellite cells around which gives the support, metabolic support and the structural support to the cell, and the nucleus is centrally placed nucleus. Centrally placed nucleus, the pseudo-unipolar or unipolar or pseudo-unipolar cells can be, neuronal cells can be seen. Either it is with a single process or without process. So the nucleus with the nucleolus can be seen at the center. Then the nucleus is covered with, the cell bodies are covered with nucleus of there is the satellite cells can be seen in relation with it. This blue dot represents the nuclei of satellite cells. So the nerve fibers or myelinated nerve fibers can be seen in bundles, which is separating the ganglia, separating the entire substance into different compartments. So this is a spinal ganglion or sensory ganglion. It can be seen like this under the microscope. So the features are, they are pseudo-unipolar or unipolar. They forms the larger neurons present in groups. That is, the collection of cell bodies can be seen in groups. The fascicles of nerve fibers passes between the unipolar neurons. The fascicles or the nerve fibers, they passes between the unipolar neurons. The nucleus with prominent nucleolus present in the center of the neuron. The nucleus is present at the center. The fascicles of nerve fibers, that is the bundles of nerve fibers, can be seen in between the neurons. The satellite cells surrounds the neurons. They are more in number. The fascicles of nerve fibers are peripheral processes of bifurcating single axon of unipolar neurons. Single axon of unipolar neurons, they are subdividing to form the fascicles. The satellite cells are there. They provide the structural and metabolic supports to the neuron and they insulate the neurons. That is the function of the satellite cells. So this is the diagram which you have to draw in the book. And next is the nerve, that is the optic nerve. The optic nerve, histology of the optic nerve, we can say that is the optic nerve can be seen with a covering of three membranes. They are the outermost dura mater, middle arachnoid mater and innermost pia mater. Optic nerve is covered with, because being optic nerve is a part of a cranial nerve, isn't it? So all the cranial nerves are covered with the three layers of meninges. The outermost layer is the dura mater, middle layer is the arachnoid matter, and the innermost layer is the pia mater. The optic nerve is covered by meninges because it develops as outpouching of the brain or the diencephalon part of the brain. There is no endoneurium, perineurium, and epineurium of the optic nerve. That is, in a normal nerve, there is the connective tissue covering named as endoneurium, perineurium, and epineurium. But in case of optic nerve, it is different. There is no any connective tissue covering. Instead, there is the meningeal coverings. There is no Schwann cells in the optic nerve. The supporting cells, which can be known as the Schwann cells, they are not in the optic nerve. Myelin sheath is produced by the oligodendrocytes in the optic nerve. Oligodendrocytes, they are the specialized cells which can be seen in the optic nerve. So that is, the myelin sheath is producing by the oligodendrocytes. Specialized neuronal cells can be seen in the optic nerve that is producing the myelin. From pia mater, a septa arises and that divides the optic. Innermost layer, innermost covering is the pia mater. From the pia mater, the septa arises and that divides the optic nerve into number of fascicles, number of small compartments. Each fascicles consist of myelinated axons of ganglion cells on the retina. Each axon is surrounded by myelin produced by the oligodendrocytes. So each fasciculus, there is a presence of myelinated axons. So this is the diagram showing the transverse section of optic nerve. It is very simple to identify. There are different covering. Three layers of meninges can be seen. Outermost dura mater. Next comes the arachnoid mater, and innermost layer is the pia mater. There is a space between the dura mater and arachnoid mater, that is the subdural space, and there is a space between the arachnoid and the pia mater, that is a subarachnoid space, and pia mater is closely adherent to the structure, and the pia mater dips into the substance to make it into different compartments. So this can be drawn like this, and at the center of this optic nerve there passes a central artery of retina and a central vein which is accompanied by it. A central artery and a central vein can be seen through the central portion of the transverse section of optic nerve. And the axons of ganglion cells, myelinated axons of ganglion cells can be seen in the compartment. So this we have to draw like with dots completely. It have to be filled with dots. So after drawing, it can be seen like this. That is, there is the nerve fascicles can be seen, the myelinated nerve fascicles, myelinated axons of ganglions of cells of retina. Ganglion cells of retina can be seen in the nerve fascicles, and they are subdivided by means of the septa which are forming from the innermost covering, that is pia mater. Outer to that there is arachnoid mater. Externalmost covering is the dura mater. At the center of the optic nerve there is a presence of one artery and vein, that is the central artery of retina, and vein can be seen. This is the diagram how we can draw it. This much of space we don't need, so that we can the space korachittu we can draw the diagram like this. Next is the last slide in the nervous system, that is the peripheral nerve. Peripheral nerve is a nerve which is outside the central nervous system. A nerve which is supplying your hand, the nerve which is supplying your leg, all the parts of the body. That is the peripheral nerve. The whole nerve is surrounded by connective tissue. I have told you three layers of connective tissue can be seen. They are named as epineurium. The whole nerve is covered by epineurium and the single fasciculus is covered by perineurium. Each nerve fiber is covered by means of endoneurium. So, a peripheral nerve, the smallest unit is a nerve fiber. That nerve fiber, each nerve fiber are covered by means of, nerve fibers are covered by means of endoneurium. And a covering of nerve fiber is endoneurium. Number of nerve fibers together forming as a fascicle. That fasciculus is covered by means of a perineurium, and number of fascicles together forming a nerve that is covered with an epineurium. Between individual axons are seen numerous nuclei of Schwann cells. Schwann cells, the supporting cells, also can be seen in relation with the individual axons. Each axon is covered with myelin sheath. Myelinated fibers can be seen along with the connective tissue. There are blood vessels, nerves and lymphatics along with the adipose tissue. Adipose tissue means that is the fat tissue. So connective tissue which is covering the peripheral nerve, there is a presence of small blood vessels which are supplying it, and nerves also can be seen to supply the walls, and lymphatics can be seen, and adipose tissue also can be seen in this connective tissue. So this is the diagram showing, this is the diagram how we have to draw it. This is the diagram showing the a section of a transverse section of a nerve, peripheral nerve. The peripheral nerve which is formed of a number of collection of neuronal fibers. So this nerve fibers, individual nerve fibers are covered with endoneurium. This rounded structures represent the cut section of fibers. That fibers are covered with a single covering that is named as the endoneurium. A number of collections of this together covered by means of the perineurium, that is a number of fibers togetherly forming into a fascicle. That fascicle is covered with perineurium. That entire nerve is covered with epineurium. Entire nerve is covered with epineurium. So in between this, in the connective tissue covering which is forming as the perineurium, or the separating the bundles, that is forming with venules, arterioles, fat cells, all these things, and small nerves. All these things can be seen in the connective tissue, that is for the support of the function of the nerve. So this is the peripheral nerve, the cut section of peripheral nerve, how it appears. This is how we can see under the microscope. We are not seeing the cut section. Under the microscope we can see it like this. The nerve cells with the what myelinated nerve fibers can be seen. Then nucleus of Schwann cells we can represent by blue dots. Then axon, these are the axons of the myelinated axons of nerve fibers can be seen, and like that in between there you can see the knots of Ranvier, the synaptic knots can be seen as the knots of Ranvier, or this are the common features of a nerve fiber, then how it appears under the microscope. So that's all about the histology of nerves, or the histology of nervous tissue, nerve fiber, ganglions and optic nerve.